Okay, good evening class. So, for tomorrow's lesson, ito na yon. Okay, so our next lesson would be gene expression. This is actually last two extension subtopic of the nucleic acid. All right, so ang scope ng midterm ninyo is all about nucleic acid, DNA and RNA, central dogma, gene expression, and application of biotechnology in refers with human health. That is our last topic actually. Okay, so let's move on with the gene expression. Yan. So gene expression, from the word gene, na-discuss na natin yan, di ba? So kapag sinabi mong gene, it is a part of the DNA, okay, or a segment. And each segment has its expression, okay? So ngayon, there are many expression of the gene, all right? So, since gene expression is very important, lalo na kapag sa development ng mga species. Okay? Kaya natin siya i-discuss. Okay? So let's start. So what is a gene expression? So gene expression is the process by which gene gets turned in a cell to transcript RNA and produce protein. Kaya nga sabi ko, di ba, kaduktong siya ng protein synthesis and central dogma. Gene expression is the process by which instructions of the DNA are converted into functional product, which is the protein. And also gene expression is the process by which information from a gene is used in the synthesis of a functional gene product. And these products are often proteins, but non-protein coding such as tRNA. What is tRNA again? Yan, transfer RNA and small nuclear RNA, snRNA. The product is a functional non-coding RNA. Okay, so the primary function of gene expression is making proteins. All right? So without protein, walang development. Hindi lumalaki ang muscles. Hindi lumalaki ang buto. Hindi ka nagkakabuhok. Wala kang melanin, mga ganyan. All right? And let's move on with the control of gene expression. How is it being controlled? Controlling gene expression is often occur by controlling transcription initiation. Okay, so meron din siyang primer. Remember, primer? Okay, the product of regulatory gene is expressed in initiates or turn on or turn off, right, in what we call the stop codon. Okay, and the turn off the expression. So sa amino acid translation, meron tayong methionine, which is the start codon. But methionine is not the initiation. Okay? Di ba remember? Meron tayong primer. Pwede siyang 5 nucleotides or 5 bases to 10 bases to start the coding process or translation process. Okay, and regulatory gene is a regulatory protein that binds DNA to either block or stimulate transcription depending on how they interact with RNA polymerase. Kasi the difference between DNA polymerase and RNA polymerase is unang-una syempre sa DNA, si DNA polymerase. Siya yung nag-pair up ng adenine, thymine, guanine, cytosine. Okay, siya ang nagbabind nun. Okay, siya ang nagbabind. Siya ang gumagawa ng phosphodiester bond. Okay? Para kada nucleotide nakadikit siya. The same thing with RNA polymerase. Sa RNA siya din ang gumagawa pero ang kapalit ng thymine is uracil. Okay? So we have let's go on with the types of genes. So we have different types of gene expression. Okay, number one is the constitutive expression. Some genes are essential and necessary for life and therefore are continually expressed. Why? Why? Kasi fetus pa lang nagbibigay ng expression. Nag-express na kagad si DNA. O ito ang dapat na itsura ng baby na yan. Okay? Yan. Until sa pagtanda. These genes are called housekeeping gene, such as actin, NGAPDH, or what we call the glyceraldehyde triphosphate dehydrogenase. Glyceraldehyde triphosphate dehydrogenase. Ganun ang basa niyan, ha? Ulit, glyceraldehyde triphosphate dehydrogenase. GAPDH. Or kapag sinabing constitutive expression is what we call the housekeeping genes. Right? And then we have the induction and repression. And then here expresses the level of some gene fluctuate in response to the external signals. So sabi dyan, gene fluctuates in response with the external signals. Ano yun? Yung mga enzymes. Okay? Yung mga enzymes na nagbibigay response. Okay? Next is let's go on with the gene expression with regards with organisms. All of our genes are present in every single cell. Okay, syempre may DNA doon. Every cell may DNA. Saan siya nakaloob? Nucleus. But not certain proteins are needed. Okay, expression of a gene at the wrong time in the wrong type of cell or in an abnormal amount can lead to deleterious phenotypes or death, or what we call the gene knockout or the knockout gene. Kapag gano'n nagkakaroon siya ng deletion. Kung hindi naman, nagkaka-knockout gene. Even when the gene itself is normal. Kaya ang case niya, pwede tayo magkaroon ng mutation. Okay. Tawag dito, mga cyst, tumors, cancers. Okay. Some gene demonstrate higher expression level once activated. This is what we call the induced expression. So, kapag sinabi mong induced, external. Okay? So, might be this is in vitro. Okay? On the other hand, some genes are repressed and their expression level are lower. It is called repressed genes. So kapag lower ang expression level ng gene, it is called repressed. Di natin siya sinasabi na suppressed, iba yun. Okay? So parang naalala nyo ba sa um, basic genetics sa dihybrid and monohybrid? Meron tayong the recessive gene and the dominant gene. Kaya nga siya de-repress kasi recessive siya. Okay, so let's go on with the exons encoding sequence. Remember that exons are the one who retains in the final messenger RNA. Introns are removed, deleted. Okay, so what is the difference between exons and coding sequences? Exons are those segments of sequence that are spliced together after introns have removed from pre-mRNA. Therefore, the coding sequence is contained in exons, but it is possible for some exons to contain on non-coding sequence. Kasi kung wala siyang promoter, okay, meron naman tayong RNA promoter and meron tayong DNA promoter. Kapag walang RNA promoter, kapag walang makitang methionine anticodons, hindi siya mag-start. Exon may contain sequence that is not translated into amino acid. These are the untranslatable gene or UTRs. Okay, with S kasi marami yon. Untranslated regions. O, kapag lalabas yan yung mga yung mga abbreviation. Mag-memorize ng mga abbreviation, mga terminologies. Okay, are found upstream and downstream in the protein coding sequences. So why do bacterial and eukaryotic cells have different mechanism of gene expression? Remember last meeting na discuss natin yung DNA replication between bacteria and eukaryotes. Almost the same lang naman sila. Okay. So why control, pumipiyok na ko, why the control of the gene expression is much more complex than eukaryotes. Complex in eukaryotes than in prokaryotes. So ito yung mga reasons behind this expression. Number one, compartmentalization of cells. Kapag sinabi mong compartmentalization of cells, kasi kasama sa gene expression yung paggagawa ng mga organelles. That is the compartment organelles. Yan. Unlike the prokaryotes, eukaryotes have different cell organelles. You see? Hindi ko pa binabasa. To perform different functions. Okay? Kasi sa prokaryotes, konti lang ang organelles. Wala din silang nucleus. So, less, what you call this one? Less ang gene expression na ginagamit. Okay? Unlike sa eukaryotes, maraming organelles. Yan. Compartmentalization allows each compartment to perform a specific function without interference from other cell function. So parang nagkakaroon sila ng, ito for mitochondria. Ito for, itong compartment na to for ribosomes, for making endoplasmic reticulum, for Golgi apparatus, and so on and so on. More extensive transcript processing. Mas extensive. So, ibig sabihin, mas crucial. Okay, regulation from the distance. Cell and tissue-specific gene expression. Larger genome size. Mas mahaba ang genome niya compare sa prokaryotes. Kasi remember, ang DNA lang nila, naka-supercoil. Kapag naka-unsupercoil siya, bilog. So, maliit lang yung genome size niya. Sa atin, sa mga eukaryotes, plants and animals, mahaba ang DNA natin. Genes scattered among the genomes. Ano ibig sabihin nga? In one genome, there are multiple genes. Okay? Ano example niyan? Meiosis. Remember? Pagdating sa metaphase stage, nagre-reshuffle sila. Nagko-crossover. And kada genome nun, may iba't ibang genes. Kaya hindi kayo magkakamukha ng mga kapatid mo. There are 4 major events that occur during the process of gene expression. Alright? So this one, for sure, hindi naman ata kayo. Nakinig ba kayo noon last time? Konti lang ata pumasok sa inyo. Okay. So ano yung 4? Transcription, RNA processing, translation, protein processing. Kapag sinabi mong RNA processing, this is also called messenger RNA. So, paano siya pinaprocess? So, gumagawa ng messenger RNA, nire-remove ang introns, nilalagyan siya ng capping. And I will discuss that later on. Okay? So, gene control in prokaryotes. Prokaryotes muna tayo. Just like sa DNA, nag-discuss tayo ng DNA translation in prokaryotes bago tayo nag-eukaryotes. So one way in which prokaryotes control gene is only one group function-related genes together so that they can regulate together. Ang tawag doon, operons. Okay? Singular name but plural in function. Excuse me. Okay. Ano bang operon? Is a group of functionally related gene that works together in prokaryotic cells. So, meron ba tayo nun? Wala. Sa ano lang yun? Sa prokaryotes, mga bacteria. Yeah. Okay. So, what are the components or consisting of mechanism of operon? We have the promoter, binding site of the RNA polymerase. Next, the operator, the repressor binding site that overlaps the promoter. Excuse me. And 3, the structural gene. Ito na naman. So as you can see here, meron tayong repressor gene, promoter, operator. And then this is the whole operon. Okay. So promoter. is a specific short sequence on DNA at which RNA polymerase attaches and initiate transcription. Diba? Para siyang related siya kay RNA, para siyang primer. Remember? And begin the transcription unit. Sa DNA, primer ang tawag. Kapag gagawa ng messenger RNA, promoter ang tawag. So kapag sinabi mong promoter sequence, these are the groups of codons that promotes for making the RNA, messenger RNA, excuse me, nare-recognize lang siya ni RNA polymerase. Okay? Yun na yun. And then operator, short sequence of DNA sequence adjacent to the structural gene that the repressor protein can bind and prevent transcription of structural gene. Kasi magkaiba kasi ang structural gene sa non-structural. Yan. So, encoded by repressor gene to regulate gene expression. Okay? So, kapag sinabi mong operator, part siya ng DNA sequence na iba siya for structural gene. Nakukuha mo, genome siya or gene siya na di ba na-discuss natin 2 types, non-structural saka structural. Kapag sinabi structural, messenger RNA, dRNA, mga ganyan. Kapag non-structural, iba yon. So ito, kapag si operator ang gagawin niya, parang sequence siya na ibang function niya to make in making the types of RNA. Kasi nga for repressor kasi siya. Activators. The activity of RNA polymerase is also regulated by interaction with accessory protein called activators. Yan, bumabalik na naman tayo sa protein. The presence of activator removes repression and transcription occur. So therefore, therefore, nag-activate siya by means of removing the repression and transcription occur. So ano yun? Una, tinatanggal niya yung nagre-repress. Tapos saka siya magta-transcript. Okay? So before the primary transcript can leave the nucleus, it is modified into various ways during RNA processing. Before the finished mRNA, the final RNA is go to the cytoplasm. Okay? So ano yung processing na yun? So from the DNA itself, annotate. So let's hindi tayo mag-annotate. Okay. So tingnan mo na lang itong aking So from the chromatin, okay, mag-unwind siya. So as you can see, this is the DNA and this is the gene. You see? Segments of DNA, gene. So ngayon, mag-i-encode siya pre-mRNA. Kapag sinabi mo pre-mRNA, meron siyang exon 1, intron A, exon 2, intron B, exon 3. Okay? Sa isang gene, meron siya. Okay? Yan. From DNA, from the gene of the DNA, meron din siyang exon and introns. Tapos, ita-transcript. Papalitan na si thymine, gagawing uracil. Okay? So, from there, magkakaroon tayo ng pre-mRNA. Okay? Na meron din exon 1, intron A, exon 2, intron B, exon 3. So, ulitin, 5' to 3'. Okay? 5' to 3' direction. Alright? And then, from there, excision. Kapag sinabi mong excision, gumagamit ng spliceosome. Okay? Excision of introns, cutting, removing, and then splicing of exosomes. Meron tayo mga enzyme that it will make it bind. Okay, spliceosomes. So, tagatanggal, tagadikit. So, ang matitira na lang si exon 1, exon 2, exon 3. And then, bago siya lalabas sa mRNA, meron tayong tinatawag na 5' and then poly A tail. And then lalabas na siya sa cytoplasm kasi sensitive si final mRNA kaya kailangan ng capping. Ang tawag natin doon, mRNA capping. So I hope you get that. Okay, enzyme in the eukaryotic nucleus modify pre-mRNA before the genetic messages are dispatched or released to the cytoplasm. At the 5' end, the pre-mRNA molecule, a modified form of guanine, is added. The 5' which function as protect mRNA from hydrolytic enzymes in the cytoplasm. Translation starts from the ribosomes. So ano yun, sir, yung 5'? It is a modified form of guanine. Okay, so puro siya guanine. Okay, yung guanine niya, magkakaroon siya ng grupo ng guanine, grupo ng mRNA na merong kakaibang guanine bases doon. Kasi modified eh. Okay, so for example, meron siyang uh, 7 codons ng mRNA. Tapos maglalagay ng puro guanine. Okay? That is the what we call the 5'. Eh naka-coil yun ha. Kasi para siyang takip. Di ba kapag sinabi mong takip, para siyang bulky. Okay? And then at the 3 end, meron tinatag ng poly-A. So ano ba itong poly-A tail? Poly-A tail, there is an enzymes that add at least minimum of 50 adenines to 250 adenines. Okay? Guanine sa 5', adenine sa poly-A tail. Kapag lumabas sa exam, multiple choice naman. Okay? Ayun po, oh. May G kasi from the word guanine. And then, poly-A tail, maraming adenine. So, RNA transcription in the linked gene to protein. Excuse me. The information content of DNA is in the form of a specific sequence of nucleotides along the DNA strands. Yan. So sabi dyan, Mendel rule. O nare-recall nyo genotype and phenotype. Actually, this is the secondary expression. Ito yung sinasabi ko primary gene expression is forming amino acids and protein. Secondary is the expression of the traits. Okay? And bridge between DNA, excuse me, and protein synthesis is RNA. So, ayan siya. So, actually, na-discuss din natin to. So, RNA, yan, messenger RNA, lalabas siya sa cytoplasm, ta-translate siya ni ribosome, and then ta-translate siya ng tRNA. Yun. Yan. DNA, transcription, gagawa ng mRNA, ita-translate siya ng tRNA, magiging amino acid or protein. So here, na-discussion natin to. Alam niyo na yun eh. Diba? So let's move on with your asynchronous activity. I have a rule here. Direction. Post your answers both on the Google Classroom discussion board that I will going to make and also in the comment section on the YouTube. Finish the video without skipping the time lapse. Kailangan tatapusin nyo. Kaya may clear lang naman yun eh. Minadali ko na nga. Each student must watch the video on YouTube per views. 1 views per student. So kung ilan ang 3A, bibilangin ko yon. Kapag hindi umabot ng 50 to 60, ibig sabihin, hindi lahat nanood. Okay, so number 1, excuse me po. What is the primary gene expression? Sabi ko lang nito, that gives vital role for body development and tissue repair. How this gene expression works in everyday life. Discuss. Yan, pwede nyo screenshot. Next, differentiate between DNA and RNA polymerase. Discuss its function. Next, what do you call the parts of the messenger RNA that stabilizes and prevent it from disintegrating in the cytoplasm? Is it also part of the gene expression? How? Discuss. So, ipopost ko to sa YouTube. Tapos yung mismong YouTube video link, yun yung ipopost ko sa Google Classroom. Okay, kapag clinic nyo yung video, doon kayo manood sa YouTube, doon kayo kung ano yung sinend nyo, pinost nyo sa Google Classroom, copy-paste nyo lang yung sagot ninyo, lalagay nyo sa comment section. Okay? So that would be 30 points. 15 points sa Google Classroom and then 15 points sa comment section. So do not forget to write your name, okay, full name and section kada post nyo sa YouTube. Kada comment nyo sa YouTube. Okay? Name, section, and then yung answers nyo as a comment. Alright? So, yan. So, thank you for listening. I hope please finish the video without skipping the ad. Okay? And the video itself. Tapusin nyo. Alright? So that's for all today. See you on next Monday. Bye-bye.